Good morning and welcome to session 2. My name's Leanne Lim and I'm an ocular oncologist here in Sydney. It's my pleasure to introduce you to the Ocular Melanoma Clinical Trials and Therapies section, where we are lucky to have Dr. Jill Hopkins with us from AOMA Biosciences, giving us an update on the COMPASS trial, as well as Dr. Amaratunga, who will be presenting an overview of evolving treatment landscapes for uveal melanoma in Australia. So I'll hand over to Dr. Hopkins. Thank you.
Great, thank you. So thank you so much. It's really a pleasure to be a part of this summit virtually. My name is Jill Hopkins, and I'm the Chief Medical Officer and Head of R&D at AOMA Biosciences. But I spent 20 years in practice in Canada, where I hail from, looking after patients as a medical retina specialist. So very near and dear to my heart are the care of patients with choroidal melanoma. And at AOMA Biosciences, we're working on a novel class of drugs, virus-like drug conjugates, which I will tell you about in detail, hoping we can really change the paradigm of treatment for patients with early-stage choroidal melanoma. I am an employee full-time at AOMA Biosciences now. And I'll take some time explaining a virus-like drug conjugate because it is a new class of drugs. And it's a very novel concept where we've taken a virus-like particle. And what's important there, this is based on an HPV virus, something like we have built the HPV vaccine for. But here we've actually deactivated the virus. So we've removed any live virus, we've removed that genetic material. And we're using the virus-like particle, the shell, the empty shell, to serve as a drug delivery. And this is critical because these virus-like particles have a real tropism for cancer cells, not normal cells, but cancer cells. So this was a discovery that came out of the National Institute of Health, the National Cancer Institute in the US, and has been researched for many years. What was very exciting and what AOMA has brought forward is the fact that you can then conjugate to the surface of this virus-like particle all of these light activatable molecules. And by doing this, we form actually a drug. So we have this little blue ball that you can see in the center with the orange light activatable molecules. That becomes the mechanism by which we destroy cancer cells. And it's a very interesting concept because it has this unique tumor selectivity that is independent of any type of genetic specific mutation in a cancer cell. So the part of the tumor cell that is targeted by this therapy are a whole sulfation pattern, GAGs they're sometimes called, or HSPGs, that present themselves on the surface of that tumor. This virus-like particle then seeks those out. We then activate that with a light, and we cause killing of the cancer cell itself. And so we showed that in multiple animal models at the National Institute of Health. We've shown over 20 different models of solid tumors that this is applicable to. And we're excited by this, particularly for early treatment, because the very targeted nature of this. So the virus-like particle only sticks to abnormal patterns on the surface of the tumor cell itself. It does not stick to normal tissue. And then it's only activated where we shine the laser light. So that dual selectivity lets us really target abnormal cells, not causing destruction to normal tissue around it. And we've now demonstrated positive clinical data in multiple early stage local cancers with a highly favorable safety profile, matching what we understand to be from our mechanism of action. So let's take a moment and talk a little bit more about the mechanism. So as I mentioned, these Belsar molecules bind to these abnormal protein patterns on the tumor cell itself, and we deliver locally in the eye via an injection around the tumor, and I'll show you that in a moment. We're also working in bladder cancer, where we go directly into the bladder and inject the tumor there. And when we activate that molecule with laser light, and this is a standard laser light, something called photodynamic therapy that has been in use for decades, actually, for the treatment of macular degeneration and for other tumors in the eye. So a very safe, well understood laser activation that causes the tumor cell to necrose, die, then and there, locally delivered. And then that sort of instigates this secondary mechanism, which is really interesting and important, we think, for long-term kind of immune surveillance. So again, you imagine that we locally disrupt the tumor right at the local area. The tumor cells then release very tumor-specific neoantigens or abnormal cells into the environment that the immune system then says, that's abnormal, I'm going to come in and get that, and starts to survey and produce then a secondary mechanism of this immune-mediated tumor cell killing. So we're very excited by that potential to have not only the direct immediate cytotoxicity, but then the longer term capability of this therapy to survey, scavenge, keep looking for those tumor cells from an immune mediated mechanism in the patient's own system. So we're hopeful with that this potential is that we'll get a long-term anti-tumor response delivered by this therapy. So we're working in several areas. Ocular melanoma, choroidal melanoma in particular, is our by far most advanced program. We're now in phase 3, and I'll talk through that as well. But we are very excited about the potential for completing that phase 3 enrollment and having a registrationable trial and treatment available for patients over the next several years. We're also exploring metastases to the choroid. So when a primary cancer from another location has spread to the choroid, using the same type of delivery and laser to treat those types of lesions. And then we're also beginning work in ocular surface cancers, melanomas, which can occur on the surface of the eye, or other ocular squamous carcinomas. We're exploring the potential there. And as I mentioned, we're active in the bladder cancer space. We're now in a 1b/2 trial based on some very compelling data from our first in-human study last year. And then as we've known from our kind of foundational work at the NIH, the potential for other solid tumors that express these types of HSPGs or GAGs on the surface. So we're very excited by the potential really as a platform, but our first and foremost area of focus is in early choroidal melanoma, where we are already in a phase 3 program. So let me share a little bit about that with you. We feel ocular oncology as a whole is a space that is, so full of high unmet need in multiple diseases. So there have been radiation as a mainstay, surgical excision as a mainstay. We're really hoping we can shift that paradigm, treat early in the diagnosis pathway, and hopefully obviate the need for larger doses of radiation or the need for radiation at all if the tumor is caught early enough, and the potential to really change the paradigm across choroidal melanoma, metastases, and the ocular surface cancers, as I mentioned. So talk a little bit about how we administer Bel-sar, particularly in the setting of choroidal melanoma. So we are excited by the potential for this to be the first drug therapy approved that has potential to preserve vision in patients with choroidal melanoma. And of course, the most important thing, local tumor control. We heard from our patients the challenges, the radiation, the things to go through. That is, of course, the most important thing. But we're excited by our phase 2 data, which I'll share with you, that shows the potential for very good preservation of vision, even when this treatment is applied in tumors that are very close to the critical vision structures, like the center part of the retina or the optic nerve. So by obviating the need for radiation in these smaller lesions, we hope to not have the radiation-related side effects. Hopefully by having something that allows much earlier treatment in the pathway, we may reduce metastases and overall safety and quality of life will be improved. So the way this is delivered, we use what's called a suprachoroidal injection, and you can see that on the slide here, the blue needle approaching the outside of the eye. So we do this in an office. We don't have to go to an operating room for this. Physicians are very comfortable with the application of a injection like this in the office setting. And we apply the Bel-sar, which is depicted by blue there, on either side of the tumor. So we put 2 injections. Those take about 2 minutes total. We give 30 minutes in between that. And then we allow time for the drug to distribute and go bind itself to those tumor cells that I talked about that are expressing these abnormal features. Then we wait about a 6-hour window before applying the light. And again, that comes from data that shows really that peak distribution in the tumor. You can think about the tumor acting as kind of a sink for Belsar. It moves into that area, gets absorbed up in that. When the tumor then has that highly distributed drug there, then we activate it with the light, again, done in the office, a standard slit lamp procedure. Many of you who have gone through treatment and eye exams will know what that looks like. And that's about a 10 to 30-minute procedure, depending on the laser map and the tumor type and location. So we are really hoping, again, that we can shift a lot of the standard of care to this in-office early treatment to make a difference for patients. So I'll walk you through our phase 2 trial, and the phase 2 means we've had a phase 1, we've shown that it's safe and overall effective. Phase 2 is then when you work with the regulators to say, right, we'd like to dose escalate. We want to understand the best possible approach that we could take to patients to explore whether this will work. And in phase 2, what we were able to do in conversation, because our safety profile has been so very good, we were able to escalate very quickly. So we started very low. So you can think about a sort of almost sham-like or no treatment-like in our very low dose regimen. So we had 20 micrograms, one laser. We moved up to double that dose, still one laser. Then we added more laser. Then we added more dose. And we saw, again, no safety concerns as we escalated up. So that we kind of called our 10 patients of subtherapeutic, meaning, we were dose escalating, we were making sure it was safe. That all looked good. And then we moved to what we would call our therapeutic regimen, where we took higher doses, 2 lasers, and then 3 cycles. So importantly, we look at this as a treatment, which up front is a sort of over a 3-month period, and then you're done. Then we follow for response to the tumor. So we looked at, did the tumor progress in any way? Was there growth along any parameter? Was visual acuity maintained or lost, and how did that look? And then, of course, looking for safety. So any treatment-related adverse events or any serious adverse events that came through in the phase 2 trial. And I'll walk you through that data now. The baseline characteristics are here. I think the key things to really focus on are the mean tumor distance. So these were tumors that were very close to vision critical structures. So usually small, early melanomas found in the back of the eye. So those that we would classify as being at a high risk for vision loss in a world where only radiation would be available to apply those. We'll then talk through tumor control. So we were very encouraged to see that at 12 months, remember we treat up front 1, 2, 3 cycles over a roughly 3-month period. Then we followed patients out to a 12-month endpoint. And we saw that in our subtherapeutic regimen, only 20% of patients had tumor control at that 12-month mark, compared to our therapeutic regimen, where 80% of patients achieved that. So that was encouraging. And I think what's important as well, and we saw it a little bit in our patient conversations just at the opening of the session, that watching for the cessation of growth, that nice scar that Professor O'Day and Georgia talked about, is really what you're aiming for. So you know that at the time you came into the trial, we did have growth that we could show in patients over time. So we knew that they were growing at a particular rate prior to the application of the Belsar treatment. Post-treatment, that leveled off entirely. So the growth was beautifully controlled in those tumors, compared to the yellow hashed line here, where you would see what the projection would have been had they not been treated. So we were very encouraged by tumor control in this phase 2 trial. And importantly, and I'll talk a bit more about it, but the visual acuity was preserved. So 90% of the patients that are now matched to what our phase 3 program is maintained vision as it was at the time they entered the trial, despite 80% of them being at that high risk of vision loss with these locations of tumors close to vision critical locations. So we did have one patient that lost vision. This is interesting in that this patient did have tumor control. So the growth was ceased in this tumor, but the patient did have some fluid around the tumor that over time spread under that central part of vision and accounted for that vision loss. No other patients experienced any vision loss. The fluctuation you see early on is really associated with the treatment over that first 3-month period, and then vision stabilized in a very nice way. And most patients came and entered the trial within about 3 letters on the chart of where they had started. So again, very encouraging for these small early lesions close to key vision structures. And then safety was also very encouraging to see. So when we look at adverse events in a clinical trial, we grade them. So grade 1 are the most minor, and they are generally self-limited. They don't cause any long-term problem for the patient. They don't require any long-term additional therapy. Grade 2, 3, 5, out through that, you can imagine become more severe, may not resolve, etc. So we were very encouraged to see that we had only grade 1 adverse events, and they were largely limited to some mild anterior, like inflammation at the front of the eye. So when you would have your eye examined over the course of your treatment program, you would see a few cells or inflammation in the eye. That all resolved either completely without therapy at all or with the use of a topical steroid drop for about 6 days. So we were pleased to see that, a very clean looking safety profile, nothing in the posterior segment of the eye, no retinal or vitreous changes. So again, very encouraged by these findings, really again demonstrating for us in a clinical setting how important that dual selectivity is in terms of where the drug binds, where the drug is activated, and then the maintenance of normal function in normal cells surrounding that. So again, very encouraging. And that allowed us to then develop our phase 3 trial. So phase 3 is hopefully the final stage of development as you take a therapy forward to bring new treatments to patients. This is one that is enrolling globally. So we are in multiple countries around the world. I'll show you that on the next slide with the map. But we are looking to enroll this in a manner that allows for regulatory registration around the world and access to patients based on this therapy. So as I say, it's kind of the final step toward getting a new therapy to come to patients. So we are busily working on this trial now. We're targeting 100 patients globally. They have small choroidal melanomas at entry. We have very clear criteria on the size and some growth to get into the trial. And we do have patients not having had prior therapy to understand the impact of our therapy. And this is what is called a randomized and masked trial. So we have 3 arms. There's the 80 microgram Belsar dose, a 40 microgram Belsar dose, and then a sham control, which is really the equivalent of a watch and wait, which many of you living in the choroidal melanoma world know that often, particularly for these small lesions, that is the discussion, conversation, decision that you may have with your physician and care team about, do we treat now or do we wait and observe, because we're not quite sure we want to head on the path of radiation and those associated side effects. So that sham arm is important in terms of being our observation or standard of care, if you will, matched arm for the watch and wait paradigm that many patients do have. And we're looking at time to tumor progression. So do we stop the tumors from growing? Do we stop them in their tracks? And what is our impact on tumor progression and visual acuity? And again, we'll have 3 cycles of treatment, that 3 months up front. We then follow for 12 months for a 15-month endpoint. So we're enrolling, and then we won't know the results of the trial until that 15-month endpoint. So still some time to go, but I think really we are very grateful to and excited by the patients and investigators around the globe. And as you can see, we're everywhere, including Australia and New Zealand. Thank you to our investigators that are helping there. And we, again, are really hoping we'll see a shift in how we might be able to approach these small early lesions for patients. There is a QR code, and then also it's just www.compasspatients.com if you want to take a look, or the QR code also will take you directly there to get more information about Compass should you desire. And I think, as I say, our focus is really in the space of ocular oncology, rare oncology, because of the huge unmet need that we see there. And the vast amount of our focus at Aura is there. As I mentioned, we're in bladder cancer as well, exploring that potential, and we'll see where the future takes us in terms of other cancers, but really hoping that we can impact ocular oncology as our first impactful area. And with that, I thank you.
Hello, And thank you for inviting Immunocore and Medison to share with you at the AOMA Summit a little bit about externally sponsored research. My name is Jennifer Johansson. I'm the Executive Director of Global Medical Affairs, overseeing evidence generation and external collaborations. And what that means, I'll explain in a little bit as I talk about evidence generation. But before I do that, let me briefly talk about the relationship, or rather the partnership that Immunocore has with Medison. And Medison is certainly well known to those of you in Australia and around the world. And their mission is to partner with companies like Immunocore. What they do is help us achieve our mission, where we want to radically improve the outcomes for patients with cancer, infectious disease, autoimmune disorders, by being pioneers and delivering these transformative immunomodulating medicines. And it's a very fruitful partnership. Now, evidence generation can take on many forms. The way in which I'm speaking to it here is from the perspective of a pharmaceutical company, or rather a biotech like Immunocore. And evidence generation in general describes the activities that really generate data across the life cycle of a particular product. As you know, this may differ if we were talking about devices or other types of registrational products. But in this case, and for keeping it simple, I'm going to talk about pharmaceuticals. So some of those aspects of evidence generation are required for registration. So if you are looking at the page here, you can see down the center aisle and to the right of the diagram, you see that encompasses things like phase 1 trials where you're figuring out a safe and effective dose, and then moving on to the other phases of clinical trials, as well as other analyses to characterize the product. The other bucket you may be more familiar with, although not familiar with it in its totality, is what we call non-registrational evidence generation. Things like real-world data generation, where you look at the use of a medication in a particular institution or within an organization. Health economic outcomes research, where you're looking at better ways to characterize the value of a product. And then there's a whole bucket called externally sponsored research. And this is basically where the sponsor of the study or the research is external to the company. That's as simple as the definition really is. So within that, there are a couple of areas that can be described in a variety of ways. But first, certainly is investigator-sponsored research, which you may be familiar with. There's academic research collaborations where we work together to understand perhaps some of the basic science or preclinical. And then there's cooperative group or large research consortium type data generation. Now, across this entire continuum, there are caveats, but high-level buckets. There are really characterizations of evidence generation that are important to meet not only the needs of healthcare external stakeholders, but those of patients. And briefly, before I share with you around externally sponsored research, I would be remiss if I didn't read to you about the tebentafusp product itself, which hopefully you're familiar with. There is a product information that you should be reading before prescribing tebentafusp, or Tebi. There are certainly important links that you can see here on this page that you could follow up at a later time. And then lastly, I do need to talk about a brief warning as it relates to Tebi, which is cytokine release syndrome, also known as CRS. This may be serious or life-threatening and can occur in patients receiving Tebi. You should only administer Tebi in an appropriate setting, and it should be monitored for at least 16 hours following each of the 3 infusions, and then is clinically indicated. And so now let's talk a little bit more about externally sponsored research as it relates to tebentafusp. So as you look at the slide here, you can see I'm really showing how data generation related to tebentafusp is across the disease journey of a particular patient. And so this starts with the primary diagnosis through their primary treatment, and then obviously, we hope, a very long surveillance period for many patients. And then as we know, some patients do recur and receive a metastatic diagnosis. So when we look to our clinical development program at Immunocore, hopefully you're familiar with our 202 pivotal trial, and that's the trial that led to the indication you have for tebentafusp or Tebi in Australia, as around the world as well. While that trial has stopped, we do continue to follow for maturity as we recently published the 3-year follow-up data and hope to bring you longer-term data in the future. So when we talk about externally sponsored research, just a caveat, this is not the whole group of research that's related to Tebi. We wanted to just show you a little snippet of it. So if we start with patients who have a primary or receive their primary diagnosis, we see Dr. Sedor at Thomas Jefferson in the United States is looking at the use of neoadjuvant therapy related to this disease state and the use of tebentafusp. And really, they're looking at whether or not patients experience adverse events and the endpoint of progression-free survival. And so before I continue with the rest of this, actually, I should just mention that publicly available information, you can see all the referencing here so that you can go in a little deeper at a later time, if that helps you. Secondly, on this side, and certainly a very large trial run by the EORTC, it is an international trial looking at the adjuvant use of TEBI in ocular melanoma. So these are patients who are at high risk for uveal melanoma or metastatic uveal melanoma who have received their definitive therapy and then receive adjuvant tebentafusp. They're looking at an endpoint of RFS or recurrence-free survival. And then skipping along in the interest of time, we have multiple trials in the metastatic setting, really trying to continue to characterize the value for patients and potentially improve outcomes. The first is a trial being run by Dr. Pilatz at the Catalan Cancer Institute. And this is looking at patients who receive neoadjuvant tebentafusp, this time prior to their liver metastasis surgery. And so here the endpoint is PCR. We have 2 ESRs, and I say we, I really meant the collective we. I should say not we because you're not the sponsor. There are 2 ESRs looking at different local regional therapies. The first is from Dr. Sidor at Thomas Jefferson, and the second is Dr. Fun at the Silver Cancer Institute in the United States, looking at the combination with different liver-directed therapy modalities. And we recognize these are important modalities for patients who have metastatic diagnoses. And so it's important to understand and characterize the safety profile as well as the effectiveness of these particular combinations. We also have another ESR similar to the TEBI-MRD one, but in this case, looking in patients with metastatic disease, usually using ctDNA as a biomarker. And then last but not least, from an ESR perspective, we have the one at the very bottom in the right-hand corner. This is one where it's being led by Dr. Joshua at St. Vincent in Sydney, Australia. And this is looking at the combination of IL-2 plus TEBI to help patients who have already recurred, unfortunately, on tebentafusp monotherapy. So these are really the bucket of all the different ESRs that are out there at various stages. The last one I actually should mention is at the very bottom, and that's a natural history study. It really is a registry that Immunocore is financially supporting that looks at the natural history of not only uveal melanoma, but also metastatic uveal melanoma. And that's being run across the world. It's an international trial. And so over time, it will better characterize the disease. And we believe it's a really important registry to be able to elucidate. So I hope this information was helpful for you, and I appreciate you letting me provide this to you as well. Have a great day.
Great. Thank you so much, Jennifer, and thank you, Medison Pharma, for that presentation. We are now very honoured to have a presentation from Dr. Aramatunga from the Alfred. This is pre-recorded. But for those of you that don't know Dr. Amaratunga, he is definitely a rising star in the field of ocular melanoma, a consultant medical oncologist, a head of molecular oncology and rare cancers at the Alfred, completed a PhD on tumor evolution in melanoma in 2023, and has a incredibly rapidly expanding portfolio in ocular melanoma and in rare cancers in general. So I'll now ask for his video to be shown, and then we will have a Q&A after the video has been completed.
My name is Mal Maratunga. I'm a medical oncologist and head of molecular oncology and rare cancer at Alfred Health. I'll be giving a talk today on uveal melanoma, the Australian treatment landscape. As an overview to this talk, I'll give a little bit of a background to ocular and uveal melanoma in particular from a medical oncologist viewpoint, and then go through the patient journey, starting the neoadjuvant space, talking a little bit about surveillance, and then the metastatic space with a focus on current treatment and what's available, as well as a particular focus on clinical trials and clinical research in this space. So from a medical oncologist viewpoint, ocular melanoma is an exciting place to be at the moment. There's been some real progress in the last few years and ongoing progress and research that we're excited to hear about. And that hasn't been the case historically. It's often been grouped with cutaneous melanoma, and we didn't see the groundbreaking advances that we saw in cutaneous melanoma and other tumor types with targeted therapies and immunotherapy. And part of the reason is that uveal melanoma is very, very different. Uh, the liver is often the predominant or only site of metastatic disease. We see late relapses, and we often see tumors that, for all intents and purposes, look very similar at the bedside or clinically, that behave very, very, very differently. And you know, part of the reason there being development and therapeutic development in the space is that we've slowly started to unpick the biology of uveal melanoma and what makes it unique and different. And we know that signaling is quite different. So unlike cutaneous melanoma, which is characterized by mutations in RAF, MEK, RAF, RAS, and NF1, we get mutations in gene AQ or gene A11, which results in activation of protein kinase C, which leads to downstream oncogenic signaling via the MAP kinase signaling cascade. And these mutations are seen in 90% of uveal melanomas. Additionally, there's been substantial progress made on risk stratification. So, separating out patients that have more aggressive or higher risk disease on a molecular rather than just a clinical basis. And that's with things like gene expression profiling, as well as analysis of specific mutations and structural variations that confer higher risk. And the other thing to note is that many of the strategies that are showing the most promise in uveal melanoma are truly first in class. So very, very different treatments compared to what we've seen in other tumor types. So looking at this patient journey, so from the time of diagnosis of a uveal melanoma, we now have a neoadjuvant treatment paradigm, which is really pioneered by Anthony Joshua, who will be present at the meeting today, and will be there during the Q&A. And he presented the NADOM trial, which demonstrated the proof of concept at ASCO last year, which is treating with darovasertib, which is a protein kinase C inhibitor, which really targets that signaling that we talked about just previously. And the goal of treatment with darovasertib, both neoadjuvant and adjuvantly, was reduction in the tumor burden with results in eye preservation. We've also seen case reports of a combination of darovasertib crizotinib showing activity, and this has culminated in the presentation of the phase 2 study last month at ESMO this year, showing very, very substantial tumor reductions in 80% of patients with eye preservation over half as a result of this treatment strategy. And we have phase 3 registration study running in many sites in Australia and overseas with significant international interest and investment, which we hope will lead to this paradigm being available for patients. Moving on to surveillance. Surveillance is a tricky space, largely because of the lack of data. In Australia, it's additionally challenged by lack of reinvestment and availability of certain imaging modalities, with a resultant inconsistency in practice. There has been a notable change in the MBS, the Medicare benefit schedule, last year, enabling a single liver MRI per annum to look for hepatic metastases in patients that have had a CT, where this may change treatment management. And one of the problems with surveillance, there's two problems. One problem is that we've got more and more treatments available, meaning that early identification of metastases, at least prima facie, may enable earlier intervention. And secondly, that there's really a lack of data on what is the best strategy. And we've recently performed a systematic review demonstrating the heterogeneity in practice and guidelines. And just as an example here, you can see the differences between UK national guidelines and the NCCN guidelines, with big differences in the modalities suggested and even options in the modalities suggested, ranging from MRI livers to ultrasounds, as well as CTs. I don't think the space has fully clarified, although there is some data to suggest that if available, liver MRI is the most sensitive treatment modality. And real world data sets are likely to really inform the practice in this space in the coming years. Moving onwards to the first line setting, and the first systemic treatment to improve overall survival in metastatic uveal melanoma is tebentafusp. And for those that you who don't know what tebentafusp is, it's an IMTAC, so immune redirecting therapy, which has an affinity enhanced HLA-A*02:01 restricted T cell receptor that's specific for the GP100 peptide and the specific components of that peptide here, fused to an anti-CD3 single chain variable fragment. And this here is the updated phase 3 data from the pivotal registration study published in 2023, showing significant improvement in overall survival with tebentafusp compared to controls, 21.6 months versus 16.9 months. And very, very interestingly, that survival benefit seems to be out of keeping with the benefits on imaging or progression-free survival, although progression-free survival or PFS is better with tebentafusp. This is the overall survival benefit is larger than one might expect with these PFS curves. This has led to reimbursement in Australia and PBS approval. And now, as we have these patients on treatment, new challenges are emerging. Things like the continuous need for weekly dosing and the convenience for these for patients, as well as the emergence of treatment resistance. And so that has justified the commencement of clinical trials, either in conjunction with tebentafusp or post-tebentafusp. And as we've seen the data, the 3-year overall survival is 27% with tebentafusp and a median PFS of only 3.4 months, suggesting a need for better treatment options. And one strategy is better combination partners. PI3K delta inhibition is a promising strategy, and PI3K delta is the predominant or an important signaling node for immunosuppressive cells, including myeloid-derived suppressor cells, M2 macrophages, and regulatory T cells. And roginolisib is a first-in-class PI3K delta inhibitor, which is actually reported substantial activity in uveal melanoma. This here is from data presented last year, showing activity, single agent activity in uveal melanoma on this waterfall plot, with a reduction in regulatory T cells, so evidence of pharmacodynamic activity in the correlative studies. And the important thing about roginolisib is this is an oral therapy that's very, very, very well tolerated with very little severe toxicity. And so because of its potential synergy with tebentafusp, it represents a promising combination partner. And we set up the TRIUMPH clinical trial, which is about to open imminently at St Vincent's in Sydney and the Alfred in Melbourne, as an investigator-initiated clinical trial. It's predominantly a safety and tolerability study, with safety as a primary endpoint, with efficacy as a secondary endpoint, and it includes correlative studies to demonstrate proof of mechanism. And so the pro forma for the study is we establish patients on tebentafusp safely, add in roginolisib, and then show that this is safe, might work, and collect correlative studies for analysis. In the post-tebentafusp setting, we have evidence for the use of IL-2 pre-treatment to reduce or polarize away from tumor-associated macrophages. And the SILVER clinical trial, which is open at St. Vincent's in Sydney and at the Alfred in Melbourne currently, it uses IL-2 to try to augment the activity of tebentafusp once resistance has emerged. And what you can see in this data published earlier this year is that the combination of IL-2 with an immune redirecting therapy results in less M2 macrophages. IL-2 has historically been very, very toxic, and so one of the unique things about the SILVER clinical trial is the use of a lower dose regimen, which ideally will reduce that toxicity burden for patients. Now, tebentafusp is only active in patients who have HLA-A*02:01, which is only 40% of patients, and for everyone else, there are no live therapies that are proven to improve overall survival. And darovasertib, which I discussed as a PKC inhibitor, combined with crizotinib, has shown very, very promising phase 2 data, which I can show you here. We've got confirmed response rates of 30%, with disease control rates of 80 to 90%, with the combination of darovasertib and crizotinib. And the combination is synthetically lethal, with crizotinib acting on bypass signaling, not pathways, when PKC is inhibited. This phase 3 study is open across multiple sites, including Adelaide, Sydney, Brisbane, Melbourne, and Perth, and is in O201 negative patients that are previously untreated and compared to dealer's choice treatment. There's also some activity in the early phase space, and this strategy is close to my heart. One target that's gaining interest in melanoma is MC1R. This is a melanocortin-1 receptor, and this is a really critical receptor that deals with the response to sunlight. So in the presence of sunlight, MC1R, and the ligand for which is alpha MSH, signals via CAMP through to CREB and activates MITIF, which is the master melanocyte and melanoma signaling molecule. And so this is a very, very obvious target that is unique to melanocytes and melanoma. A first-in-class theranostic, VMT01, has presented safety data and some early signals of activity earlier this year, and an alpha-9 3408 study has recently opened at the Alfred in Melbourne, Fiona Stanley and Genesis Care in Perth, and will shortly be open at Peter Mac and the Austin in Melbourne, and takes all melanoma, not just uveal melanoma. So this is an interesting new strategy as well. There are also several other early phase options available that allow uveal melanoma, including ONM-501, which is an intradermal STING agonist available at several Australian sites, and BNT326, a HER3 ADC at St. Vincent's in Sydney. And I'll stop it there.
Great. Thank you very much, Mel. We've now got some time for question and answer from anyone. And also, there are some questions in the chat. Leanne, do you want to? There are certainly some questions for Jill from some of the ophthalmology group. So maybe Leanne, if you would moderate those.
Sure. So we have a question from Rodo Day asking Jill, do you have longer term control data for the initial cohort that had 80% control at 1 year?
We do not. So as a small biotech, that was when the endpoint completed. We do have some patients that entered a registry where we can follow for METs and overall survival, but that was a sort of voluntary enrollment for patients. So there are gaps in that data, but some of that will be available as feasible, but we don't have long-term reliable measurements, for example, or visual acuity. From our phase 3 program, however, we are doing a 5-year long-term follow-up where we will have ongoing reading center-based ultrasound measurements of the tumor, visual acuity, and of course, safety, metastases, and survival. So the phase 3 program will be very important that patients do enroll on into that long-term follow-up, because that will be very important long-term data with a new modality like this.
Great. And we also have a question from one of our ophthalmologists, Chanel Sharma, who asked about the patient you described who had presumably macular edema. Were they given any anti-VEGF treatment?
They were not, I believe, on study. So they had fluid associated with their tumor. And as I mentioned, the tumor stopped progressing, stopped growing, but the fluid did sort of transect into that foveal region. But an important question, of course, anti-VEGF being an important part of ongoing control, but not in this case.
I guess as a follow-up to that question, did you include subfoveal tumors, and do you laser the fovea?
Yeah, it's a great question. So we do, when we apply the 2 laser treatments, we allow the foveal edge to be well treated with the first treatment and then avoid treatment in the second one. So we are mindful of that. But to date, certainly in phase 2, we didn't see any secondary RPE changes or vision loss associated with tumors in that foveal region. But we'll monitor that very closely, of course, in phase 3 as well.
Okay. Because I guess with photodynamic therapy also used in like other pathologies like CSCR, there have been described kind of choroidal changes. So just wondering if that has been seen in this treatment.
Yeah, particularly not when using the suprachoroidal delivery. So our very first trial, our phase 1 first in human trial, used intravitreal injection of Belsar, which was the only available way to inject into the eye at that point in time. And we did see more, still an overall favorable benefit risk profile that allowed us to go to phase 2. But interestingly, in intravitreal, we did see more inflammation in the back of the eye, posterior segment inflammation. And we did have a couple of cases that had vision loss associated with that sort of RPE, that epitheliopathy, if you will, that we see sometimes. So it was interesting, the sort of odd good things that come out of COVID, for example. We were shut down for a while. Suprachoroidal delivery was really coming along, and we were able to explore using the time we couldn't be in human clinical trials. That's when we explored our animal models with suprachoroidal compared to intravitreal, and saw much better uptake in the suprachoroidal with suprachoroidal delivery in the tumor itself. And then our phase 2 clinical program, we saw none of those side effects that emerged with intravitreal. So the suprachoroidal delivery may be just the sort of sweet spot of getting a drug where you want, being able to then laser in a way that we have not seen associated epithelial or RPE changes. In phase 3, we are masked, so we are, we don't have data at this point to know, but certainly we haven't been seeing SAEs emerging in a masked fashion even. So that's encouraging.
Oh. And I have a last question for you. Do you think that, I know that this is for the treatment of small choroidal melanomas, but do you think that there will be a role for maybe looking at larger tumors, or?
Yeah, we do. And I answer that for a couple of reasons. One is that, certainly in our bladder cancer. So again, the ocular oncology space was our first foray. But we did have some really striking data that came out of NIH in bladder tumors. So some people are like, how did you get from the eye to the bladder? Well, we had really good preclinical data. And so we took, honestly, the ocular dose and volume, small, and we injected them into tumors in the bladder that were 10x the size of any melanoma that's in our current trial. And strikingly, the idea of that trial was it was patients that were scheduled to have their tumors resected anyway. They would have their Belsar treatment up front, and then the surgeon would take them for their scheduled resection of the tumor 7 to 14 days later. And in our intermediate risk bladder patients, 4 out of 5 of those had no residual tumor when the doctors went to resect them at that 7 to 14 day window. So that was striking and interesting evidence of that mechanism in terms of the very early tumor necrosis. We were then fortunate, though, to be able to get histopathologic samples from those bladders that showed all of that secondary immune mechanism that I mentioned, all the infiltration of innate and adaptive immunity cells. So that gives us a lot of sort of in human confidence that larger tumors could be impacted. Again, why we were interested in being in this early phase to start was that that's an area where there's so much uncertainty about how to manage them, right? Particularly a foveal tumor where radiation is the only option, and there's really no other development in that space. So we felt like the largest unmet need was there, and worked with the regulators around the globe to have that trial design supporting that with the observation arm or sham arm in the trial. But we absolutely think about ways to expand into larger tumors.
Amazing.
Well, the inclusion criteria for the phase 3 trial, maybe just emphasizing that again. What are the criteria for entry into the current enrolling study?
Yeah, so there's small lesions up to 2.5 millimeters in thickness and 11 millimeters in largest basal diameter. And they do have to be untreated with anything in the past, and no evidence of metastatic disease. And then we do have evidence of documented growth as one of the criteria. And the reason that's important, although it makes waiting and following the patients, and we have great sites in Australia who've been doing a beautiful job getting patients in, but the time to show that growth is important. But that's key for actually the efficiency of the trial design, right? So we knew in our phase 2 trial, we had that as a criteria as well. Patients that were actively growing in the sham-like arm, by 6 to 9 months, they had crossed that set progression threshold. So that allows us to have a shorter endpoint, a smaller sample size, in fact, and hopefully bring to patients sooner what might be meaningful. Our anticipation, though, is once available, that documented growth would not be something you would have to show. You as ocular oncologists and in discussion with your patients would say, this is a lesion I have some concerns about. I can treat you today up front. I'm not concerned about vision loss with this therapy. We'll follow you on. So it helps that sort of dilemma around the potential radiation associated complications. So growth is important for getting into the trial. It only has to be a little bit of growth, but showing some documented growth we thought was important to make sure that it's not a lesion that won't show growth in a reasonable time frame within the trial for us to understand our endpoints.
Great. And from a medical oncologist point of view, is there any thought to giving them with checkpoint inhibitors, particularly in the bladder?
Yeah, great question. So yes, we would see this as being very synergistic with checkpoint inhibitors. And again, with our safety profile, I mean, that idea, if you treat locally upfront when the tumor is there, safety profile looks great, induce some long-term immune surveillance, you could then go on to add additional therapies. And absolutely, in some of our preclinical animal models, we saw tremendous synergy with a checkpoint inhibition approach.
Yes, for those patients who are online, a checkpoint inhibitor is a drug which takes the brakes off the immune system. The immune system, when it gets turned on in cancer, has accelerators and brakes. And the big breakthrough in immune therapy for cancer has been to realize if you take the brakes away, the immune system can actually work quite well to attack many cancers. And these drugs have become standard of care for most common cancers now, actually, in various stages. So when we hear about treatments which activate the immune system, then the next question from most medical oncologists is, why didn't you add in a checkpoint inhibitor? So,
fair question.
So that's well, that's good to hear that that might be in the pipeline. Certainly, bladder cancer is another area of need, and would be fascinating to undertake. So there was a question about the COMPASS trial in terms of sites in Australia. I don't know if you have that list available.
We do. So we have Rod O'Day, who spoke earlier today. Melbourne is a site for COMPASS. Bill Glasson in Brisbane. And we have also Riaz Biku in New Zealand.
Great. All right. Because we should see if we can get a site up in Sydney, but sounds fascinating.
Working on it.
Yeah.
I don't know if there was any questions in the chat about the talks from Medicine or from Dr. Aramatunga. Mal is unavailable to take questions. I can certainly field any if there are. But I don't see any at the moment. There certainly are exciting advances in the treatment of ocular melanoma into the future, and I anticipate that we will have effective systemic therapies in the years ahead. And I'm not talking many years, I'm talking a few years from the various companies involved for metastatic treatments and indeed also for localized disease. So I guess I'm very hopeful that we will never have to do enucleations into the future, and we're working very hard to achieve that goal. And we have, and more importantly, vision preservation, which is an incredibly overlooked endpoint in to date, because we haven't had the luxury of having treatments that work. So once you've got a treatment that works, just to make sure that the vision as a result of plaque radiotherapy doesn't suffer, and indeed maybe even we can move away from plaque therapy altogether if we've got treatments like Belsar that can be effective. So I don't see any more questions. Jill, so what is the timeline for the end of the study?
We'll hope to have our top line data in 2027. So still a bit of time to go. It's the length of the endpoint, but important again in showing that long-term cessation of growth.
And when does accrual, you don't have to, obviously, that may be confidential, but accrual is progressing presumably, and the top line data is an endpoint at
15 months.
15 months. Okay.
Yeah.
So that 2027 is not that far away, really.
It always feels long for patients, I know, but again, I think, the hope is that with this trial, and again, we're in 17 different countries around the world for, access on the heels of this registrational program. So.
And obviously being a Canadian, and also I'm waving the also Canadian citizen, presumably you've got sites peppered around Canada as well?
Indeed.
Yes.
Absolutely.
Right, great. So, and I guess in relationship to the previous talk from Medicine, are you at the stage yet where you're entertaining investigator-initiated proposals for other
Yeah,
ideas?
it's a great question. I think we've already had reach outs for them. I think, our yes, I would say, but we're still early. I think with that registrational program ongoing, I mean, Immunocore has the advantage of having their approved product and availability for patients, which then allows a lot more access for IISs. But we've had a lot of reach out from our community. I mean, again, the engagement from the ocular oncology community for our platform and therapeutic potential has been huge. And we get, all kinds of like, this would work great in ocular cancers. What if I did this cycle, or what could I retreat? Like, if the safety looks that good, what about treating down the road? So I think our current approach is let's get this trial, executed, get that data, have that, solid evidence in hand. And then I would anticipate lots of questions that we could dive in on with the community.
I think there is a question, Anthony, from Catherine Hahn about Triumph and Silver trials. Can you see that one on the...
Yeah, but can I just finish with, I'll just finish with Jill.
Sorry.
With solid tumors, have you done any intratumoral injections? I'm just thinking in terms of liver metastases.
Yeah, it's a fascinating thought also. So it was intratumoral in our bladder program. We injected into the tumor. When we think about exploring ocular surface cancers, we would also think of an intratumoral or peritumoral approach. And yes, it was interesting. I was actually sitting at a meeting about a year ago now with someone from Unicorn. They're like, "Have you thought about injecting into a liver met?" I'm like, well, but our metastases to the choroid trial will be interesting there, because there we'll have, it will be indirect, but if it's lung or prostate or breast that has metastasized the eye, and we see a good response in that. And again, based on our tumor models out of NIH, we saw response over 20 different tumor models. So yeah, the intratumoral, I mean, that's kind of the exciting thing. And when you think about how sort of med tech and the approaches, people are like, well, you could only get where you could, inject and laser, but now we can start to get just about anywhere, lung, GI, even in the brain, people are doing photodynamic therapy and glioblastoma with probes. So the potential, I think, for something like this has really expanded as we see the ability for visualization, robotic surgery even to reach places that we wouldn't have before. So a lot of interest, I think, in what that intratumoral approach could be, particularly in, ocular oncology. Again, docs do injections and lasers all the time. This fit right in their flow, as did urologists, cystoscopies and lasers. But gastrointestinal would be another area that you could really see potential, where docs scope and laser and snip and cut all the time. And again, if you treat up front with a safe opportunity, you could go on and do other things, but see how the patient responded. So it's a very interesting platform.
Great. So just thanks for that. I think we're sort of wrapping up, but Dr. Catherine Hahn from Auckland has a question about the TRIUMPH and SILVER studies. So just to remind people, the TRIUMPH study is first-line tebentafusp plus ROGI, and that is first-line. So, and that tebentafusp is supplied through the PBS, because it's a standard of care therapy. The patients do unfortunately have to have a Medicare card, or I imagine potentially pay for it. And then the SILVER study also is on the PBS, because the patients, when you're taking tebentafusp, for those that don't know, usually it is unclear when you should stop, because probably the tebentafusp has an effect on slowing down tumor growth. So we often will continue a patient certainly beyond the first scan where the tumor appears to be growing, and that's the window when we have them on a PBS drug, so reimbursed government drug, and so that government drug continues intercurrently with the IL-2. So so unfortunately at the moment they are restricted to patients where someone else is paying for the tebentafusp, i.e. at the moment that's the Australian government. But happy to talk about if there's any other mechanisms into the future as well. But certainly an area of interest. There may be other studies that Mal mentioned where those caveats may not apply. I think he mentioned a radiopharmaceutical study and a HER3 ADC, which seem very promising, and there may be more flexibility enrolling New Zealand patients. So I think we're more or less at time at 12:55 Sydney time. And now we will move over to our gold sponsor presentation. So thank you very much, Jill. Thank you, Mal, for your contributions. Our gold sponsor is Idea Biosciences. This is a presentation from Dennis Chang. For those of you that don't know, Idea Biosciences is a precision oncology company who are advancing a very broad and exciting pipeline of targeted therapies for cancer, which target cancer drivers in particular, and certainly have a very ambitious and really exciting momentum in the space. Now, the drug we're talking about today is darovasertib, which is a PKC inhibitor, which Mal mentioned in his talk. And I'm sure that Dennis will talk about that. but the company does have other assets that target various drivers in cancer, targeting, for example, MTAP-deleted tumors, a thing called a PARG-G inhibitor, and a DLL3 antibody-drug conjugate, which also looks like it's got fantastic activity. I've been working closely with IDEA for a long time. They're a very exciting company. I very much appreciate their involvement in the AOMA Summit, and we really couldn't do it without them, quite literally.
I would first like to thank the organizers for inviting me to speak today. My name is Darren Beaupre, and I'm the Chief Medical Officer at IDEA Biosciences. What I hope to do in the next 5 minutes is give you a brief overview of our organization, as well as update you on some emerging data from our lead program, darovasertib, in uveal melanoma. IDEA Biosciences is a company that is approximately 10 years old, which focuses on harnessing synthetic lethality to deliver precision medicine therapies. We have multiple pipeline products that are all first-in-class in various stages of clinical development. Utilizing deep understanding of the molecular characteristics of tumors, we apply monotherapy and combination treatment strategies to elicit profound anti-tumor effects in various tumor indications. This slide highlights the breadth of our pipeline, the various stages of clinical development, and the important collaborations we have with key industry partners. Suffice it to say, we cut across all phases of clinical development, and we have partnerships with some of the leading pharmaceutical companies in the world. Currently, our lead program is darovasertib, and shown here is the biology behind why this product has had such profound activity, either as monotherapy or in combination with crizotinib in uveal melanoma. The vast majority of patients with uveal melanoma have mutations in the genes GNAQ or GNA11 that drive activation of protein kinase C. This pathway is truly the harbinger of the disease. Darovasertib is a protein kinase C inhibitor that can effectively inhibit this pathway, which is highly relevant in the primary uveal melanoma setting. In metastatic uveal melanoma, it has also been found that overexpression of c-Met is relevant to the development of metastases, and is why a combination of darovasertib and crizotinib, a c-Met inhibitor, is used in this setting. Based on the biology of the disease and the data that we have seen thus far clinically, we believe darovasertib can impact the entire patient journey of subjects with uveal melanoma. In the primary uveal melanoma setting, neoadjuvant darovasertib can induce tumor shrinkage, which can lead to eye salvage, as well as improvements in vision. For those subjects at risk for metastases post-primary therapy, the darovasertib crizotinib combination may additionally prevent future metastases. This combination is also relevant to subjects who present with distant metastases as a treatment for advanced stage disease. And importantly, all of these phases of the disease can be treated effectively, irrespective of the subject's HLA-A2 positive status. The data that supports these assertions I will cover at a high level over the next few slides. Here we provide data from the recent Society of Melanoma meeting that was held in Amsterdam in October. We presented data from our phase 2 study in untreated subjects with metastatic uveal melanoma, revealing an overall response rate of 34%, a duration of response of 9 months, a progression-free survival of 7 months, and an overall survival of 21.1 months after treatment with the darovasertib and crizotinib combination. All of this was achieved with a manageable safety profile. These results are notable since they reveal a response rate and progression-free survival that are more than double current standard of care agents, and the overall survival of 21 months was significantly greater than the 10 to 12 months that has been reported for this patient population in several meta-analyses. On the neoadjuvant side, we have also presented data at the European Society of Medical Oncology meeting, also in October. In this slide shows the majority of patients treated with darovasertib monotherapy prior to primary local therapy have tumor shrinkage. And importantly, in those subjects destined for enucleation, nearly 60% of eyes were saved. In those subjects requiring plaque brachytherapy, we also saw a clinically significant reduction in radiation dose to key visual structures, and also the predicted risk of 2200 vision or worse, that is legal blindness, at 3 years was reduced in 2/3 of treated patients. Also, we provided evidence that vision could be improved even during the neoadjuvant therapy in both patients with large tumors requiring enucleation and in those with smaller tumors requiring plaque brachytherapy. An average of 3 lines of vision was gained in the enucleation subjects and 2 lines for the plaque brachytherapy group. In summary, based on this remarkable activity as both monotherapy and in combination, darovasertib has received breakthrough therapy designation from the FDA, and 2 registration trials are currently underway in both the neoadjuvant and metastatic setting to confirm these initial findings. In addition, an adjuvant trial is in planning. To conclude, IDEA Biosciences is in the business of bringing first-in-class transformative medicines to patients in need, and we believe with darovasertib we have taken our first step towards this ambitious goal. With that, I thank you for your attention and the privilege of providing this darovasertib data update.
So that brings us to the end of session 2. It was a wonderful session on our ocular melanoma trials, the local and metastatic disease approaches, and thank you very much to our speakers and also to our sponsors and for all of the interesting questions. So that brings us to lunch and we'll reconvene for session 3. So thank you all.